Welcome, everyone. I'm Beth Daly. I'm the executive editor and general manager of The Conversation. Thank you for joining us today on this incredibly important subject on the different stages of developing drugs and how AI is playing a role at each of those steps. It's a fascinating area. It's an important area, but it's also can be a complicated area. So we have what we do best at The Conversation is to explain things in plain language to you. And I'm thrilled about that. First, some housekeeping things. If you have a question, please do not use the chat. No one will see it. Please put it in the Q&A. We have someone moderating the Q&A and we'll be feeding questions to Eric. What we'll do, we'll start first with a conversation and then we'll open up for questions, probably somewhere between 30 minutes to 35 minutes in, depending how the conversation goes. I did also want to say, for those who are new to us, a little bit about The Conversation. We're a national nonprofit newsroom, although we also have local editions now. And our secret sauce is pairing expert editors who really know their stuff, and I'll introduce Eric in a second, and he's one of those people, with experts based at universities and researchers. And they collaborate on writing a story. It's very rigorous, and it goes out. We publish it on our website, which is wonderful. We publish about 10 to 12 stories a day. But the real power is that all our content is Creative Commons, meaning anyone, it's always free for anyone to read and republish. So, last Thursday we were in 1,312 news outlets, and that's about par. So, and we're the most read nonprofit news outlet in America as of recently. And I'm obligated to say, but also happy to say, if you like what you hear today and what you read, we are in the middle of our end of the year donation campaign, and we would love support if it feels right to you and you can afford it. So, without further ado, I'm so pleased to welcome Eric Smalley, the moderator for today. Eric is our science and technology editor. He's over 30 years of experience as a science and technology reporter and editor. His work has appeared in the Boston Globe, CNET, Discover, Nature, Science, Scientific American, Wired. He even co-founded and edited Technology Research News, a weekly online news publication that covered university research in emerging technology and applied science. He's also he's also unrelated to this, but also fascinating, a very, very talented musician, and has a great band. So I'll pass it over to you, Eric.
Well, thanks,
I'm
Beth.
going to turn, yeah.
Okay. So I want to introduce our speakers and just sort of reiterate that the topic here is AI in biomedical research and drug discovery. And AI is such a huge area. It is so hot. When we try to break it down, it may seem like we're focusing but even AI in biomedical research and drug discovery is a huge area. And we're going to kind of go some high level kind of look at what this means. But there's just so much going on here so that if anything sparks your interest, I encourage you to kind of dig in further because there's so much going on here. So joining us today is Jeffrey Skolnick, who is Regents Professor, Marian Mazie Gibson Chair, and Georgia Research Alliance Eminent Scholar in Computational Systems Biology at the Georgia Institute of Technology, aka Georgia Tech. He is the director of the Center for the Study of Systems Biology in the School of Biological Sciences. He's also the chief scientific officer of the Ovarian Cancer Institute. He is a fellow of the American Association for the Advancement of Science, the Biophysical Society, and the St. Louis Academy of Science. He has developed AI-based approaches to predict protein structure and function with applications to drug discovery and finding off-target uses of existing drugs for aging, cancer, chronic fatigue syndrome, and precision medicine. Also joining us is Benjamin P. Brown, who's an assistant professor in the Department of Pharmacology at Vanderbilt University, where he's also a member of the Center for AI and Protein Dynamics. His lab works on creating new computer-based tools that make drug discovery faster and more reliable. Their projects combine ideas from modern machine learning, older but still widely used chemical data methods, mathematics, and large-scale computing. They use these approaches to better understand how proteins and other molecules behave, to speed up the search for promising drug candidates, and to build models that can help guide the development of new therapies. So let's start with kind of the big picture. What are the stakes? How is AI changing biomedical research and drug discovery? and what is the potential we're talking about? Jeff, let's start with you.
Thank you, Eric. Well, I mean, the upside potentially is very, very large because, as one may or may not know, one of the frustrating things about drug discovery is because the problem is very complicated, success rates are very low. I mean, 1 in 5 drugs is going to fail safety. So, and then of the ones that pass safety, a significant fraction, roughly half, don't work. So what this is telling you is despite of the fact that the people who are doing it are very intelligent and have done an extraordinarily good job, you have key issues that you want to do. One is, how can you predict which is a target which is driving a particular disease? And then once you've identified this target, how can you guarantee it's A, going to work, and B, if it's going to work, it isn't also simultaneously going to kill you? So these are very outstanding problems in drug discovery. And AI can play a very important, though not 100% guaranteed role. I mean, this is an emerging field. And, you know, amongst the things it can do is it can actually help you figure out, well, what are the driver proteins or proteins for a given condition? Is it simply one, or is the condition driven by multiple diseases, in which case you have to deal with the issues of polypharmacology? And AI is really good because it can look at almost everything and look at what's so-called higher order terms, basically correlations between the two. Okay, imagine you're juggling one ball, it's easy. Now you got to juggle 50,000 balls and figure out where's the red one and the green one, because those are the ones your eyes have to focus on. That's the problem. And then once you've identified the target that you believe to be a key driver of the disease, then you actually have to figure out what is the best technique to modify the target. And not only do you want to do this, you want to not only hit the target that you care about, but you also don't want to hit the targets you don't care about, because you don't want side effects and adverse interactions. And again, here is a perfect place where AI can play a very important role. And there's various types of AI you can use. For example, you can ask, can you predict, as in the AlphaFold 2 and its various derivatives, can we predict the protein structure? Can we use this to predict what are drivers of diseases? Can we include genetic information? Can we include disease trajectories? And as you start to include this plethora of information, which is actually informing what you want to do, this is where AI, large language models, can play a very important role, because unlike us, where we have a finite bandwidth, AI can basically look at all available knowledge, and on a good day, it can identify either known and weak connections that are true. And on a bad day, it does what's called hallucinating. It sees things that are weak and probably false. So it can provide an accelerated approach, because at the end of the day, what do you want to do? There's a lot of diseases which don't have a cure. Most diseases actually contemporarily in drug discovery are maintained. You have high cholesterol. You take a cholesterol drug for the rest of your life. You never get it fixed. You have an autoimmune disease. You're almost always maintaining things, not fixing them. If you're lucky and you have a strep throat, an antibiotic will cure you. But for most diseases, it's maintenance. And there are so many diseases that are not cured. Moreover, you may have a treatment like in cancer, and it buys you 5 years, and now you're in stage 4 and you've exhausted all the standard of care drugs. Now what do you do? Here AI can play a role to suggest alternatives and options when there are none. And again, you have to do this carefully. You have to be skeptical, because it can lie to you, and it's very convincing, or it can tell you the truth. And so part of the art in the game is developing these tools, and the fields are developing at a very, very rapid rate now. We're kind of in the exponential growth phase, and it holds a lot of promise, but one also should be careful not to overpromise. It will help, it will accelerate. It is not a substitute yet for real experiments, real clinical validation and trials, but it can reduce the number of things you have to do. And that's where you really matters. Because suppose rather than having to screen millions of molecules, you could screen 10. And suppose not having a hit rate for safety of 4 out of 5, it was 99 out of 100. Or efficacy, if you could shift the statistics. This can be translated both into economic savings, but more importantly, into helping improve the lives of people. So it has a lot of promise, but it still has to be developed. And there's tremendous efforts across many fields with many techniques. There's a lot of players in various different approaches, and it'll be very exciting to see what actually plays out, and what's going to dominate, what's going to be the ultimate dominant, and what's going to be the ultimate payoff. But it is certainly a very powerful tool when used appropriately with appropriate controls and validation.
Okay, we'll get into a couple of areas, more specific areas of how AI is being used. But let's back up and give some basic explanations or descriptions, or basic definitions here. Ben, first, when we say, when most of us use the word drug, we're actually talking about a wide range of therapies. So it's not just one thing. Can you very briefly sort of explain kind of the range? So we've got small molecule drugs, biologics, gene therapies, cell therapies. Briefly, what do those mean?
Yeah, that's a great question. And so, as some folks might know, we have these things in our bodies called proteins, and proteins are fairly large molecules. You can sort of think about them as machines that carry out specific functions and interact with one another. And oftentimes when we're trying to treat disease, what we're really trying to do is alter the function of specific proteins by turning them down, turning them up, changing the way they interact with their partners. And so fundamentally, drugs don't have to just interact with proteins, but a major way in which our sort of current repertoire of medications work is by doing something to the functions of these proteins. And so then, when you think about what is a drug, oftentimes it's something that is a small molecule, much, much, much smaller than a protein, 1/100th the size of a protein, something very small, but that can fit into it and change its function. So these are things like, aspirin and Tylenol. These are small molecules. There are other things, slightly bigger, basically other proteins. So the best example of this is antibodies. When you have, a vaccine and your body develops antibodies to then go target, some viral antigen. So your body has a set of instructions to learn how to target something. What you're doing is you're creating these larger functional molecules to go and interact with proteins in a different way. And so these modalities, small molecule or protein and antibody, are very different types of molecules. They have very different scales and very different rules. And so the way that you approach designing them, discovering them is very, very different. And then gene therapy naturally is even sort of a larger step beyond that. There's a lot of different ways you can start to modify the genome or modify expression of certain proteins. So the scales vary widely, both in the size of the molecules themselves, as well as sort of the systemic effects or the effects they'll have throughout the body.
Got it. And Jeff, when we say AI in this context, we're mostly, I think, talking about deep learning, right? So can you sort of very briefly explain the concept of artificial neural networks and what machine learning means and what the deep in deep learning means?
Okay, so so originally, as in, last year's Nobel Prize, in physics, it went back to people started trying to understand how neurons worked. And so what you basically had was you wanted to build what's essentially a network. So you have a whole bunch of inputs, which are stimuli, and a whole bunch of outputs. And then there's a little brain. The little brain could have been one node. And they have different weights, just kind of like your brain actually works with the axons and the connections and puts them together. God bless the gamers, because the gamers created GPUs, and GPUs are incredibly good at manipulating matrices.
Those are the chips, the graphics processing units.
The graphic processing chips. Basically, what they originally did was you wanted to have an image which was really high resolution, and you want to rotate it. What did it do? It rotated a matrix. That's the mathematical process. So it was set up. So what you can then do is to say, well, I don't want just one neuron. I want a collection of neurons, because the neurons can have reinforcement learning. And so you could do 1, 2, 3. Eventually, you could do thousands of them. Okay. And then, of course, this was just simple connections. And then you can do things like have very sophisticated neurons like transformers, which do specific things. It's like a special purpose little tool. Okay. It learns things. And then you can add things in deep learning, like not every input variable is important. Okay. Some may be really important and some may be noise. So there's a thing called attention, which is basically a very fast way of saying, I ought to pay attention to this one because I believe this one is important. And this little guy over here, it's basically not doing anything. And so that's what neural networks you can do. So super neural networks with transformers, this is deep learning. And they have literally now billions, if not a trillion parameters. Okay. Good news, they can learn because all these things are connected. Higher order correlations between events. What this means is, suppose you have a property which depends on the properties of 5 things simultaneously. It's not just simply A alone and B alone. A plus B is different than A alone and B alone. Now, A, B, C, D, E, F, and G. So you have a set of conditional things. And a lot of physical interactions are conditional. So it can learn these patterns. Okay. And it learns these patterns. And this is the good news. The bad news is, if I have a trillion parameters, it's like the old joke with enough parameters, you could fit an elephant. Okay. So you got to be really careful that the size of the thing you're training on and testing on are sufficiently large that it isn't going to simply memorize everything, in which case it's one hell of a sophisticated database. And you can use these things as databases really fast because it learns everything and it gives you the answer. You know, you don't have to ask the question to figure it out. That's one thing. And then, of course, there's these large language models which try to learn, again, in a different way, context. Okay. When you have a sentence, okay, and this was a very important thing. Okay. If you say, are A and B related? Okay. And whether this is a pair of words or a series of amino acids or a set of genes, the first thing you could do is to say, well, you know, if I look at a sentence and they're immediately next to each other, A is not B, that's easy. Now, suppose you had a paragraph which was 500 words long, and at the end of it, it said, oh, by the way, A is not related to B. You have to include all these correlations, and you have to understand the context. That's what these large language models have the capacity to do. Okay? And of course, they're kind of like complementary things. And again, what they tend to do is they tend to learn on examples, and they have some capacity to generalize. But you got to be careful. Are they simply learning this body of data and A looks like B? And as long as A and B have subtle but well-determined relationships, it can tell it. But then there's the stuff which is outside the box, which is never seen. And this is where the gold is right now in AI. You want it to actually do something which is completely out of the box. It's not in the training set. Like an example would be, this is 1905, and Einstein says that the speed of light's a constant. There was no way in the state of physics that you would have guessed that the speed of light should be a constant. You might have known that the speed that had certain properties. This was a leap of faith, or gravity distorts space-time. Okay, these are leaps of faith that are not currently available, but this is another area of research which you really want to do. And it's actually not just relevant for understanding how brains work and being creative. It's also relevant because embedded in all this data, all this biological data of now petabytes and petabytes of data, are these higher order correlations which are there, which are going to suggest new things. It's not only A looks like B, and it's very sophisticated and very subtle. And if A and B, the connections are strong enough and right, you call it insight. If it's strong and weak and wrong, you call it an hallucination. So you got to be careful. It can do both. And so you want to check them with multiple AI approaches. It's kind of like, if this consensus is truth and beauty when you don't know what else you're doing, okay? The idea being that they've learned on different things, and if they learn the same things independently, the probability is that they're true. So that's kind of how these things work. So what you need to do this is you need a massive training set. So for example, AlphaFold had millions of sequences and 100,000, couple of 100,000 instructions, perfect for deep learning. In general,
Just say that AlphaFold is...
It's an algorithm which predicts protein structure, the three-dimensional biologically active form of a protein. A protein is a polymer, and a polymer can have many, many shapes. It's like a piece of string. The ones you care about, it will fold up, and this is the miracle. A subset of them, they have a sequence, that is to say a string of amino acids arranged in an order. A subset of them fold to very well-defined structures reproducibly, and it's these structures which has been indicated or do the business end. And they may have more than one conformation, and they have dynamics that you have to worry about. But to zeroth order, you can map the sequence, which was the protein folding problem, to the structure. There was enough data to do that. Also, what helped is the library of single domain proteins. Proteins kind of come in like little pearl necklaces. You know, there's these little individual spherical domains of a couple of hundred residues that kind of quasi-fold independently. There's only about 2,000 of them, so it could learn them, okay? And then it could map the interactions. So again, A looks like B. And then it can be really much more sophisticated. You don't realize it looks like B, but that's how it works. So that's kind of, to first order, what these things do. You're looking for analogy and generalization. Whether this is reading an article and saying, tell me what the latest news is and what's the best stock to buy, same kind of technology. Or tell me, based on this pattern, what is the best small molecule I should design that sticks to this thing and acts, either turns it on, it acts like a cork, or induces some conformational transition, turns stuff on and turns stuff off. It learns a pattern. That's what it does.
Okay.
Very sophisticated. But
yes.
that's in essence what these things do.
Right. So AlphaFold is the model. It's developed by DeepMind.
That's correct.
Yeah.
Of which they won a Nobel Prize last year,
right,
along with Baker.
Yeah. So Ben, how is this technology being used kind of in the front end of this whole thing when we think about biomedical research to understand molecular dynamics, and the kind of the pathways, as researchers call them, the biological processes that are involved in health and disease? What is the role of AI in studying those things?
Yeah, thank you for that question. It's a really interested and sort of multifaceted one, and so I think one of the first things to mention is what Jeff pointed out early, target identification. And so you have a disease, and we want to know what types of what do we want to do to a person to treat that disease, and this usually revolves around first identifying, well, what are the proteins or the little molecular machines that we can manipulate in order to give some type of positive effect for disease. And so target identification is a really important and really big topic right now. The second sort of part of this is saying, okay, well, how do we actually design the medicines? And so, AI and deep learning are large right now, but it's worth mentioning that, for the last decade and a half, people have been using much smaller machine learning algorithms to help them sift through chemical information and try to come up with like what compounds might be more interesting than other compounds if I'm trying to design a drug for this. And so a lot of the ideas, where you could use machine learning to help you identify, small molecules for targeting a particular molecular machine to treat a disease are not new and have been in practice for a while. And they're very useful. And so then, the idea would be, hopefully, now that we have more data, emerging ways of thinking about deep learning, we can make them increasingly useful. And so you think about target identification, helping come up with good molecules that have the type of bioactivity that we're interested in. Does it turn it off the way we want to, et cetera. And then sort of the third thing, which is, sort of where molecules go to die, is like, drug metabolism and pharmacokinetics. And so the idea that you put a molecule into a person, how fast is it excreted? How long does it stay in its functionally active form? It turns out that people who are really, really good at making molecules, one of the hardest things for them is to not necessarily make molecules that have the right bioactivity, that do the right function, but it's that have the right bioactivity and do, the right function while also being stable and safe and not excreted in the human body. And so that's a challenging problem because it's the data, well, there's a lot of reasons why, and we can talk about that later if you want, but so these are sort of, three areas, and then you can extend it to more modern, drug design, like with proteins and antibodies and et cetera. There are similar challenges. I think the really exciting stuff, especially with, John Jumper and Demis Hassabis's work on AlphaFold and the rest of that team, and the group obviously at the Institute for Protein Design and University of Washington, designing these technologies to help people predict protein structures and design proteins, is we've sort of changed, I think, how we think about a lot of these problems. In 2013, when there was a Nobel Prize for molecular dynamics simulations, so computational tools to help you understand the motions of molecules as they move according to physics. That there's a whole huge body of scientific research built around those ideas, and I think the really exciting thing right now is the demonstrated success of tools like AlphaFold have really changed the way people think about approaching these problems and given people sort of like this new this new repertoire of approaches that they can use to start to think about drug discovery, and that's very exciting.
Well, and Jeff, let's go a little further with that. Can you give us a sense of what today's AI technology has done in terms of the scope of these things? So in terms of, there's a huge range of possible molecules and molecule dynamics. There are all these compounds to screen. Are we talking about the equivalent of like strapping on a booster pack on scientists and just letting them do a whole bunch more? Or is there something like qualitatively different, like things that we couldn't do before that we can do now? And what's, like from say 20 years ago to now, what's that kind of scale of change in this process?
I would say the biggest scale of change, which allows you to do, and again, it's kind of like Ben has alluded to. You have a target, so you got to worry, what is the individual target doing? Okay. But a cell is like a crowded party on New Year's Eve. There's a lot of molecules interacting, and if you've ever seen, David Goodsel's beautiful pictures of what the interior of a cell looks like, it's Times Square on New Year's Eve, packed. Okay, so there's a lot of intermolecular interactions. And a lot of these interactions, some are just adventitious and accidental, some are true and actually biologically relevant. And so it allows you to start to, first of all, deal with and integrate going beyond the individual molecules to the collective behavior of the molecules. I mean, we really would like things to be like a Swiss watch model. You have a gear, you fix the gear. I mean, and if you have a disease, which is a Mendelian disease, it's one gene causes the disease, like sickle cell anemia. That's great. And that's a perfect target, and it is for gene therapy. You fix the mutation, which causes the disease, and life is good, assuming that you can selectively target enough of it without off-target interactions. But a lot of diseases, like cancers, are caused by a collection of malfunctioning beasts. And so how do you deal with it? It now allows us to start to think about the so-called polypharmacology, the interactions of the players of the targets that are driving the disease. And so it's not just one and a zero. This one is the only thing and everyone else is true. It's a lot of things that are contributing of differential weights. What drives the disease? Can you understand how diseases, even on this level, are related to each other? You could imagine, and it's affords you the possibility not only of just simply being a tool. This is the thing, it allows you to think conceptually about how these things are organized. Like you could imagine every disease is a special case, right? I got this disease, it's unrelated to that disease, except that there's one little ugly fact or a virtuous fact. Diseases tend to be comorbid. They tend to co-occur. So for example, if you have hyperthyroidism, you are very, very likely to develop Alzheimer's. Okay? Kind of weird, right? And then there's all sorts of really weird stuff that you'll see that tends to co-occur. Why is this? When you can integrate and look at the collective behavior, which is what AI allows you to do, because it can look at this, we can only look at pieces. It can then say, well, these have common drivers, and this allows you to construct disease interrelationships. This can then offer the possibility in drug discovery of broad spectrum treatments rather than narrow spectrum treatments. It's like an antibiotic you could treat. It allows you to deal with the issue of, you have an antibiotic for one bacteria, okay? And that's great. You just spent $1 billion and you got one bacteria. You want them all. Well, if you understand the disease interrelationship, which is now hoping to do, we can actually treat whole collections of these things. It can also, and this is really important apropos of this, allow you to understand, and this is another component of AI and drug discovery, which is not unrelated. All of us has about 1.5 million people, and it has disease trajectories. So diseases tend to occur where you have this position 1, position 2, position 3. You have disease 1, it gives you disease 2, which gives you disease 3, which therefore suggests that if you were to go back to the root of this simple disease to treat, you may be able to stop a whole bunch of stuff. You couldn't do this before. And how do you analyze millions of trajectories and millions of data? I mean, you can't do it without a tool to do it. So it's a way of allowing you to think in aggregate, okay? Similarly, for drug discovery, I want to target this molecule. It gives you possibly better tools for designing molecules that are more likely to be specific. and it offers the possibility of worrying about all the other molecules all the proteins it's going to interact with due to the off-target interactions, which are causing the side effects. A lot of drugs fail because of QT prolongation. You really don't want your heart to pace properly. That's a real showstopper. Okay, so if it messes up this ion channel, in addition to cures you, what do you want? Okay. Right. And so this allows you to do this. It changes the vision. But again, what I would argue, and I think Ben raised a very interesting point, we don't want to forget the power of the existing tools that were pre-AI. They have their place and can contribute as well and are sometimes more appropriate. Because if you have tiny data sets, AI isn't going to work. It's simply going to memorize. Okay. Right. On the other hand, sometimes what you have is relatively small data sets, and then more traditional tools might be more appropriate. What I would argue is when you're looking at AI in any class of machine learning algorithms, what is the best tool to solve the problem? If all you have is a hammer, everything looks like a nail. If you have a toolkit, you can have a fine tweezers, or you can have a saw, and you pick and choose the tools that are appropriate for the problem and the dragon you want to slay. And we're building more tools. So basically, we're fleshing out the toolkit. And so it's on many levels, it can change what you do. But the key thing is, and I think this is an important point that I want to emphasize, it is still not a substitute for doing real experiments and real people. I wish it were. Build a virtual human, an avatar. It's got all your genetic variations, and one day the dream is this will happen. We're not there yet. But right. What it can do, and this is important, is it can prioritize more shots on goal. It tells you where to look, as opposed to all things being equal, you have to look everywhere. Right? There were screening tools, and a lot of medicinal chemists have tremendous biochemical intuition. It can be like a medicinal chemist in a box to some extent. It has a lot of this intuition built in.
Right. We're going to get to questions from the audience in a minute, but Ben, once the process doesn't end once you've found the compound that you're pretty sure is going to have the effect you want, and you're pretty sure isn't going to cause a lot of harm. There's a lot more that goes on in getting a drug into the hands of your doctor, right? So there's a whole bunch of testing. There's preclinical testing in animals, typically mice, right? There's the clinical trials where you're testing in humans, phase 1, 2, 3, and beyond that. There's even after drugs on the market, you're kind of studying how it's doing and how people are doing. Is there a role for AI in this, in helping with all of these levels of testing?
Yeah, that's another really interesting question. So I think part of this, if we take a step back, we keep using, AI or machine learning, and really that's sort of a description of just like they're approximators for complex, like nonlinear functions, I should say, not complex. And so, the idea is that you can shape them in different ways to do different things, right? A machine learning model or an AI is not like one thing. And so for some tasks like image recognition, you've got one type of neural network that is built to see images and interpret them a particular way. For another type of neural network, you've got something that has sort of a deep intuition for geometry. So that way it has certain rules baked in and it knows what to look at and focus on. And maybe that's how, we help protein structures and things like this. And so there are some models that are very general purpose and some models that are very special. And I think when we have problems like, let's say, maybe we wanted to simulate a clinical trial or figure out, what patients should we include in a clinical trial based on their EMR data or things like this. You can build models to do that. You can build a model for anything. I think the hard part is in figuring out, well, like, how do I build a model to sort of intelligently sift through these data and meaningfully combine them, so you're not just sort of fitting data, to fit data. But yeah, I think it'd be really exciting to, it's really expensive to run a clinical trial, and I don't run clinical trials, in full disclosure, right? But it would be nice if you could do things like, compare to cohorts of people that are outside of your current trial to understand whether or not, this is having an effect. So you could get away with enrolling fewer people. Or it'd be nice to know, what are the people that you should enroll through looking at medical records. So there are a lot of ways you can imagine, taking something that has done well in an animal and seeing if you can get it, through trials faster by leveraging your data just more effectively. The other thing we talked about is safety profiling. And so is there a way to learn a general relationship between like a molecular structure and like how likely it is to be excreted, for example. But anytime you get into this, it's sort of the closer you are to causality, the better your model is going to be. So in other words, if you look at something that does image recognition, right, it's got a picture of your face, and your identity is attached to your face. Like it's sort of like there's a relationship between you as a person and like the pixel representation that is your face. And so it's very tightly coupled there, right? Now you take something and you say, I want to take this molecular structure and know whether or not this is going to have a half-life of, however many hours or days. Now those things aren't necessarily coupled because it's correlation. You've got the structure, and then there's several intermediate steps that are potentially, variable that we're not modeling explicitly. And so the best we can do is correlate to some extent, this output. And so I think when we think about like what type of data are we trying to fit, and then it helps us sort of think about like, what are my expectations for how good this is going to work, and where can I really apply it in a way that I think is going to be generalizable. So the closer to causality we can get when we build our models, I think the happier we're going to be.
All right. I'm going to turn to some audience questions. And this first one, Jeff, you were alluding to something like this. The question is, could you discuss the use of digital twins of cells to predict drug efficacy? And digital twins are these sort of simulations on steroids of a system, and it can be anything from a cell, as this person refers to, or a city. But in terms of cells and drug efficacy, is there a use for digital twins, and what's the relationship between a digital twin and an AI model?
The answer is, in principle, yes, and there's a tremendous amount of activity now in the field of trying to build as accurate as possible digital twins, virtual cells, so to speak. And I would say the field, as a matter of practice, is still in its infancy because, you got to get started somewhere. It's just like the Wright brothers plane. I mean, initially you fly a couple of 100 feet long before you go to the moon, okay? But the promise is that it could do it. But of course, you got to be a little careful in your statement. Is this a generic average population digital twin, or is it the digital twin of you? Okay. Now, initially, what you may want to do is kind of an average digital twin, because you want to, if you're going to build this stuff, it may well be easy to do an average rather than the idiosyncratic behavior of an individual. We're actually in the process now of literally just doing this. We're putting in everything we possibly can, kind of alluding to what Ben, mechanistic information, all available data on T cells, trying to predict, patient-specific T cell exhaustion and mechanism processes. This is just one example. Some people look at just differential gene expression data. My own personal feeling is you got to put in as much information of the cell as possible in order to make it as accurate as possible, because these things interact in a collective. You take one drug, and there's this L1000 database, and you say, how many genes levels change which they're making proteins due to one drug, just one, what drug on average, it's just 1 millimeter perturbation, hundreds. So it's a collective effect that you have to worry about. And so, yes, it's happening, and I think it will happen, in stages, just like everything. I mean, there kind of be like initial frustration and successes, but this is the goal. And of course, if you have a digital twin of a cell, is that enough? Because, a lot of complex diseases involve the coupling, say, of your immune system, the neuronal system. Cancer is a typical example. A lot of cancer cells actually are wired into axons, okay? And some neurotransmitters cause cancer cells to grow. And your microbiome couples to your brain and stimulates it. So the question is, because we have on the order of 10 to the 37 cells in our body, even when we were to be successful in a petri dish, kind of a virtual cell, a cell line model of a digital thing, that doesn't necessarily mean it's going to tell us how this thing works when we want to work about the organism. Because cell line studies, have cured cancer many times, but they're not characteristic of the actual disease in many people. Sometimes they are, sometimes they're not. But you got to start somewhere. You can't build a a digital avatar yet of everybody or even cells, but it's going to happen. But you got to start, as with most things, a lot of these things are reductionist. You start with an elemental piece. You try to understand how it works, what doesn't, how do you fix it, how do you make it as good as possible. Then you put another one and you let them interact with each other. And it's kind of like a buildup, which is kind of how a lot of science, but not all science, happens. And I think that it will prove to be immensely powerful. But it's going to be over a period of years. It's not going to be yesterday, is I think a fair statement.
Okay, good. All right, next question. Ben, you were kind of alluding to or talking about data and having the right data in terms of being able to get at causality. This question is, what kind of data is a bottleneck for AI drug development?
Yeah, that's a great question. I'd say right now, there's a lot of interest in trying to get to that last part of, so for small molecule, drug development, I think there's a lot of traction in being able to say like, oh, can I predict what this drug does? Can I rank how important it is in my list of potential molecules? And really, there's data that's required, but it's also a bigger issue is just the representations. Where there's not a lot of data are in things like the toxicity, the drug metabolism, the pharmacokinetics. And so there are some open consortiums like the Open AdMet group that's trying to sort of make data available to people so they can train on. There are pharmaceutical companies right now that are trying to come up with different deals to have biotech companies share data. But this is the challenge because again, if and when I talk to medicinal chemists, they have thousands and thousands of molecules that do exactly what they want when you're just looking at the bioactivity. But then you look at what fraction of those compounds actually do what you want and then have the right sort of toxicity, metabolism, et cetera, profile. And it's a very small fraction. And so how do we sort of design up front for those molecules or search spaces of chemistry that are interesting that already have those sort of good profiles? And so it would really be nice to have more data in that space. But I also think there's a lot of publicly available data on various things in drug discovery that's just underutilized, and sometimes we just have to think about how do we represent our system a little bit better so it learns the relationships that we want it to learn. And not everything should just be fed into a language model. Not everything should just be fed into a convolutional model. Not everything should just be fed into a graph. And so we can be a little bit more creative too about how we force it to learn things.
And following up on that, there's another question that's kind of not exactly the flip side, but from a different angle, and that is the issue of data privacy. Is any of this data coming from people's medical records, and are there, supposedly anonymized data sets from this? And and how do people, or is there issues of consent and privacy involved in any point here?
Yeah, I can't speak to this from like a first person perspective because I don't work with EMR, electronic medical record data. But from what I do understand from colleagues in that space, I mean, there's de-identified patient data banks, right? And so there's never an attachment of specific identifying, numbers and names and that kind of stuff from a person to a collection of data about them when you're using them for these algorithms. There's also patient consent. There's waivers to consent and things like that. And so Vanderbilt has been sort of operating in this space for a long time. And so if you're interested in this stuff, I really do recommend looking at some of the other work that's going on at Vanderbilt University Medical Center in the human genetics and other areas. Because they thought about this a lot in designing their electronic medical record and also in trying to prioritize patients that they think could really benefit from certain therapies.
All right. And Jeff, there's another question that has to do with when data is sparse. There's not a lot of data that you need of a certain type. The question is, are computational chemistry simulations being used to create synthetic data? We had an article recently kind of explaining what synthetic data is. Is that something necessary? Is there a need for synthetic data or other ways of making up for a sparsity of data?
That's an excellent question. So the answer is yes to both. Okay, so I mean, you can do computational chemistry, and as long as you're reasonably sure that the computational chemistry is generating at least a reasonably biophysically accurate description of the system, then this data is very useful. Sometimes because of just defects in the force fields and stuff, it may generate artifacts as well. So you always worry when you're going to optimize on something, it's going to find the pits. Okay. And the pits may be, in the real world is a set of additional constraints that you don't know about. You mimic it with this set of data. And then when you optimize it, you don't have these constraints, you may get alternative solutions, which are simply non-physical and not relevant. So you got to be a little careful about that. But yes, the second thing is, as Ben kind of alluded to, not every different AI methods are appropriate for different classes and sizes of problems. Our experience is if you have moderate sized quantities of data on the order of thousands, not hundreds of thousands to millions to trillions, you're much better off using algorithms like XGBoost, random forest models. They tend to generalize much better and less memorization. Now, this is an empirical observation. I've argued with my CS colleagues. What I would really love you guys to do is to tell me the class of algorithm which is going to be perfect for this particular problem. Okay. I have this algorithm. If it has these features, it's like a recipe. Apply this and it's likely to work. Empirically, what I can tell you happens in the real world is people will try 10 different machine learning methods. And you try your best to separate the training and the testing so you don't memorize the answer, because then it's just a database. It's garbage in and garbage out, because you want it to generalize. And you see, use various statistical measures like how, what's the false positive rate, what's the true positive rate, so on and so forth, rock curves or whatever. Not important what they are. But the point is, and then you say method X, for God knows why, is the best for this class of problems. And you try it again. So you have to tune the approach that you're doing, which is kind of what you indicated, to the size of the data at hand. And it may well be that there are problems of data sets which are sufficiently sparse that even XGBoost doesn't have enough data. But then you can use these so-called, going back to the old ideas of protein folding, these knowledge-based statistical potentials. They're not perfect, but they're better than nothing. So you have to pick the problem and the solution you have to the size of the data that you can actually generalize and train on. Otherwise, you're simply going to memorize, and what you're going to do is you're going to have ungodly results for data which is in the training set, and then when you give it something extrinsic, it doesn't work. So a lot of this is just empirical trial and error in essence. There is no algorithmic recipe yet which tells you, for this structure, this is what you ought to do. There's some heuristics that we know about by just, you do enough of this. Eventually it's trial and error, and you start to figure out patterns.
All right. And another question we have is wondering, given that the number of potentially promising correlations increase as these data sets expand, does that create an increased temptation, as this person asks, to act in haste and pursue development paths with limited oversight, given competition in the market? Is there a need for oversight or applying, a way to apply the brakes on that? And I'll throw that to Ben first, but you can both jump in on that if you have thoughts on that.
Yeah, I'm not sure I totally understand the question. I think, any clarification?
Sure. I guess the idea is that given you have so many possibilities, and I guess this gets to whether this is a problem at all, given the AI technologies, is there a risk that there's a kind of a rush to a potential solution that maybe you shouldn't have pursued, given all of this huge potential and number of possibilities?
I see. I think if I'm understanding this correctly, particularly the bit about the correlations, I think there's always a risk that as there's more data and there are more correlations between things, that you produce a model that you're very excited about, that you think works really well, but maybe it doesn't work very well because you haven't tried to break your model hard enough. And I do think that in a perfect world, that's the kind of thing that peer review would sort of suss out, and we'd be able to say, we've thoroughly vetted something, and this is definitely good or this is not. I think I think the field is still growing to accommodate this. Right now, it seems to be more of a community effort where somebody will put out a tool and then the community will sort of evaluate it through their own trial and error and sort of come to terms with, is this a tool that I can use for things, in either a general sense or even for if it's just a narrow sort of application, does it work in my narrow application? And then, either yes or no, and people use it or not use it. And so yeah, there's definitely a big problem, and Jeff's alluded to this a number of times with building models that are generalizable. And I think the responsible thing to do as a scientist, right, is to try really hard to break your own models, right? So before you don't want other people to break them, you want to sort of break them and know where they break. So that way, when you put them out, you can be like, hey guys, this is what I learned. This is what the model's good for, and this is where it breaks. I think if we try to divorce ourselves a little bit from the excitement and some of the hype in the space and just be honest scientists, that's what we should be doing, right?
Okay. All right. Well, we're getting near to the end of our time. I've got a last question, and a questioner asked a very similar one, so I'm going to give one version to one of you and the other version to the other. So Ben, I'll start with you. So we've seen AI brought huge changes into biomedical research and drug development. As the technology gets more powerful, more sophisticated, where do things go from here?
Yeah. Where do things go from here? I mean, honestly, right now, I don't quite know where things go or how fast they get there. I think it's the world is sort of shifting under our feet in a way that's very exciting, but also a bit unpredictable, and so we have to stay a bit nimble. The way that I guess my lab is thinking about this, is on two fronts, right? For drug discovery, what are the pain points, right? There's been a lot of progress that's been made over the last 30 years in developing computational tools to help with small molecule drug design. More recently, now we can start to do protein design, but for small molecules and peptides, the question is, what's really going to be the breakthrough that makes it so that way you can screen 1 billion molecules in a day on your computer and then you get your hits, and then really the amount of medicinal chemistry optimization that has to occur is substantially lower. So we're really, sort of interested in understanding what that breakthrough will look like, and it'll probably be a combination of old things that people have been developing with some new insights from the AI field. We're also really interested in things like what were the paradigm shifts, in our field and structural biology. So when you're thinking about molecular machines, understanding how they move, which tells you about their function, and then being able to, now predict their structures from just a sequence. These were two monumental sort of shifts in the field with molecular dynamics simulations and AlphaFold. So what does that next sort of third thing look like? What goes beyond MD simulations? And I think this is going to get into sort of connecting the atom level resolution or similar sort of resolution with large biological processes occurring at the cellular or multicellular level. And so what does the technology have to look like in order for us to get that type of resolution? So I think the next 10 years, a lot of people are going to be pushing in that direction.
Okay. And Jeff, I'm going to give you their question, the reader's question, viewer's question. What would the next AlphaFold moment look like, and where will we be in 10 years? And it's a bit unfair. It's asking you to predict what you can't really predict, but how would you address that question?
I mean, I'm probably going to be wrong. So let me start off with that. I mean, because, I suspect what we're going to be able to do is to understand and predict the network behavior of cells within processes between cells, and at some level go from genotype to phenotype. It won't be perfect, but it will be collective and it will be robust. And operationally, what you're probably going to see is more drugs approved with a better safety profile, even though not perfect, and more efficacious. And you may well also see different approaches emerging as a consequence of the tools as one starts to understand this better treatment of algorithms that can deal with this network behavior, which I think the polypharmacology is very important for a lot of biological processes. A lot of things happen, and also, what I think one's going to see integrated is we're going to see much more. People talk about omics, but you really want to be able to treat it from a bottom up of the whole collection, because, Sitting in you is a quarter of a million different types of metabolites. The body isn't making these things because they're junk, okay? It's expending a lot of energy. They're clearly involved in doing something, probably regulation, control. How do we harness all this information, build tools that can integrate this and are truly predictive, both in a collective sense and an individual sense? Because at the end of the day, what you really care about, and possibly this will start to happen within the next 10 years, is this is you. These are your cells in you. I know this information complemented by a subset of experimental data. These are the drugs that are going to work, and these are the drugs that are not going to work. And for a subset of intractable diseases, hopefully they'll be less intractable as a result of these collective network-based tools delivering on their promise. That's what I would expect to see. And you'll see an integration. Bottom line is it's, you can study these fields as silos. Nature doesn't know about silos. I mean, nature doesn't know how we divided up the fields in the late 1800s. It's collective. There's a lot of stuff going on. You need to know the physics, the chemistry, the biology, the genetics, all the omics, and you need to put this in and have a way to think about this and deal with it in a rigorous, predictive way that can be tested and is generalizable. And I think that's what's going to happen.
Well, it does sound promising. And I think the big picture of, incredibly exciting possibilities, but lots of caveats, and that it really is not going to be a silver bullet, and you still need the human mind involved here. So I want to thank Jeff Skolnick of Georgia Tech, Ben Brown of Vanderbilt. This has been really informative. I hope the audience got as much out of this as I did. And I'll pass it back to you, Beth.
Yeah, thank you so much. That's totally fascinating. And for me, it was what I got it. It was well explained. I feel like we covered a lot. So I'm really grateful to both of you and to you, Eric, for coming up and taking us on this journey. Thank you all again. If you have any questions or any ideas for future webinars, please reach out to us. We're easy to find. Just Google the conversation. You'll find emails. Again, we are a national nonprofit news outlet. And so if you care about us and have the means, we would appreciate the support. In the meanwhile, have a wonderful Wednesday afternoon. And thanks for joining us.
Thank you
so much for having us.
Yeah. Take care.